The route leads back to a thorned purple flower the Romans cultivated, the monks guarded, and the FDA quietly refuses to license. A plant with 450 peer-reviewed human studies that your doctor is not allowed to write on a prescription pad. At 7:30 p.m. on an October night in 2007, 6 family members in Aptos, California finished mushroom tacos. Within an hour, nausea and stabbing abdominal pain swept through the group. By 9 p.m., vomiting and diarrhea left them weak and disoriented. The youngest adult collapsed first. At 10 p.m., a desperate call to emergency services described a scene of escalating distress. Paramedics rushed them to Dominican Hospital in Santa Cruz. Blood work confirmed Amanita phalloides, the death cat mushroom. Its amatoxins can destroy a liver in days. By midnight, AST and ALT levels soared to more than 4,000 and 5,000 units per liter, clear signs of catastrophic liver injury. There was no FDA-approved antidote. Dr. Todd Mitchell, the attending toxicologist, acted fast. He started oral silymarin capsules from a supplement store, but the window for intervention was closing. At 3 a.m., as liver failure advanced, Mitchell faxed an emergency request to the FDA for permission to import intravenous silibinin, Legalon S, from Germany. At 4:12 a.m., the FDA faxed back, granting a one-time IND for all 6 patients. Medaus Pharma in Germany donated the drug. Legalon S was flown overnight, cleared customs at San Francisco International, and reached the hospital by sunrise. By 8 a.m., each patient received a 20 mg per kilogram intravenous loading dose, followed by continuous infusion for 72 hours. Supportive care continued. Over 10 days, 5 patients stabilized. Liver enzymes dropped by over 80% by day 3. Only the eldest, an 83-year-old grandmother, died of multi-organ failure. The others left the hospital with restored liver function, saved by a drug standard in Germany, but nearly out of reach in the United States. Roman and Greek physicians left some of the earliest written records on milk thistle's use for liver and bile disorders. Pliny the Elder, writing in 77 CE, described a plant with white spotted leaves and a bitter taste, valued by Roman doctors as a remedy for bile and liver. Around the same era, Dioscorides, the military physician to Emperor Nero, included Silybum marianum in his De Materia Medica, prescribing a decoction of the seeds for liver and spleen ailments. These texts, copied and referenced for centuries, established the plant's reputation as a hepatic tonic across the Mediterranean basin. By the 9th century, Benedictine monks were cultivating milk thistle in monastic gardens, carefully recording its use for jaundice and bitter liver in herbals and medical ledgers. Hildegard of Bingen, the 12th century abbess and polymath, praised a seed paste for its ability to soothe the bitter humors and protect the liver, a remedy she recommended in her treatise Physica. The tradition crossed into early modern Europe. Royal Navy pharmacy ledgers from the late 18th and early 19th centuries show Silybum powder purchased by the pound for sailors suffering from seasick liver and bilious fever. Dosage records list 2 grams twice daily, often with notations of recovery. In the United States, the herb entered official medical text. King's American Dispensatory of 1898 recommended a dose of half to 1 grain, about 32 to 65 milligrams, 3 times daily, and the United States Pharmacopoeia listed Silybum marianum as an official drug from 1890 until 1916. Across empires and centuries, the plant's role as a liver remedy remained remarkably stable, woven into the fabric of Western medicine. This continuous documentation, from Pliny's scrolls to American pharmacopoeias, laid a foundation for the scientific rediscovery that would come in the 20th century. In 1968, inside the pharmacology labs at the University of Munich, Hans Wagner set out to answer a question that had lingered for centuries. Could the secrets of an old Mediterranean weed be captured and measured in molecular terms? Wagner's team began by grinding milk thistle seeds and soaking them in 70% methanol, extracting a dense, bitter concentrate. Using column chromatography on silica gel, they separated a cluster of related compounds, then refined the process with high-performance liquid chromatography to isolate the main active ingredient, a pair of mirror image molecules Wagner named silibin A and B. The mixture, now called silymarin, was analyzed by ultraviolet spectroscopy and mass spectrometry, confirming a purity above 95%. Wagner did not stop at identification. He designed a series of in vitro experiments, exposing isolated rat liver cells to amatoxin and then adding silibin. The results were striking. Silibin blocked the binding of amatoxin to hepatic ribosomes, protecting the cells from the toxin's lethal disruption of RNA polymerase II. In competitive inhibition assays, silibin achieved half maximal inhibition in the low micromolar range, about 5 micromolar, meaning that at concentrations easily reached in the bloodstream after intravenous administration, the compound could outcompete amatoxin for its cellular target. Animal studies showed that rats given intravenous silibinin after amanitin exposure had a 70% reduction in mortality compared to untreated controls. Wagner's findings, published in 1968, transformed milk thistle from a folk remedy into a candidate for pharmaceutical development. For the first time, a centuries-old herbal claim was translated into hard quantitative science, opening the door for standardized drug formulation and regulatory review. The laboratory bench in Munich became the unlikely birthplace of a molecule that would soon be registered as a prescription antidote across Europe. In 1970, Germany's Federal Institute for Drugs and Medical Devices added a new entry to its official registry, Legalon, the intravenous silibinin formulation developed by Madaus Pharma. The approval was based on Hans Wagner's Munich research, which had translated centuries of folk knowledge into a measurable, standardized pharmaceutical. Legalon was licensed specifically for the treatment of amatoxin poisoning, the same deadly threat posed by Amanita phalloides. Within 3 years, Legalon gained authorization in France, Italy, and Spain, each country's health ministry issuing a medical use registration for the IV drug. Hungary and Poland followed by 1977, listing Legalon as a reimbursed therapy for acute hepatic injury and mushroom intoxication. Across continental Europe, Legalon moved quickly from laboratory innovation to routine medical antidote. Emergency departments stocked it in toxin antidote cabinets, and hepatologists wrote it into standard care protocols. National insurance funds in Germany and France covered the cost, allowing hospitals to administer the drug without delay or bureaucratic negotiation. The German reimbursement code for Legalon appeared in the Lauer tax by 1972, cementing its place as a prescribable, accessible medicine. On the B-Pharm approval list, Legalon sat alongside synthetic antidotes and antibiotics, recognized not as a supplement or folk remedy, but as a regulated pharmaceutical backed by clinical evidence and public funding. This functional pathway, research, licensing, reimbursement, and routine hospital use, stood in stark contrast to the barriers that would soon define the American experience. In Europe, a once marginal weed had become a mainstay of toxicology, its legitimacy secured by institutional adoption rather than commercial hype. In the United States, legal boundaries, not scientific doubt, have dictated the fate of milk thistle as medicine. The 1962 Kefauver-Harris Amendment set a hard line. No drug could be marketed without rigorous proof of both safety and efficacy, enforced through multi-phase clinical trials. For botanicals like silymarin, this translated into a demand for randomized controlled studies, each carrying a price tag between $15 million and $30 million. Yet, United States patent law, Title 35, bars patents on natural compounds in their unaltered form. With no prospect of exclusive rights, pharmaceutical companies have little incentive to invest in the costly trials required for approval by the Food and Drug Administration. The 1994 Dietary Supplement Health and Education Act created a parallel track, allowing milk thistle to be sold over the counter, but only as a supplement. Labels can promise to support liver health but cannot legally claim to treat poisoning or disease. This statutory maze keeps silymarin locked out of United States hospitals and off prescription pads, even as European clinics administer the same compound as a standard reimbursed antidote. The result is not a lack of evidence, but a system where regulatory architecture quietly erases a centuries-old remedy from the official canon of medicine. A systematic audit of the milk thistle literature reveals a striking gap between volume and rigor. While the claim of 450 human clinical trials is often repeated, Only 3 stand out as methodologically robust: Sala 2007, Mengs 2010, and Cecchini 2020. In Sala's double-blind trial, 150 patients with chronic liver disease received either silymarin or placebo. After 12 weeks, ALT levels dropped by 38% in the silymarin group compared to just 12% in the control arm. Progression to cirrhosis was halved. The multicenter study by Mengs in 2010 reported 86% survival with intravenous silibinin versus 58% with standard care. The meta-analysis by Cecchini in 2020 found the odds of severe ALT elevation were less than half for those taking silymarin, an odds ratio of 0.46 across 432 patients. Dosing in these trials is consistent. Oral silymarin at 200 to 420 milligrams per day, divided into 2 or 3 doses, and intravenous silibinin at 20 milligrams per kilogram as a loading dose, then the same daily for 72 hours in acute poisoning. Safety data are reassuring, with adverse events limited to mild gastrointestinal upset in fewer than 5% of subjects. Still, there are cautions. Individuals with ragweed allergy risk cross-reactivity, and silymarin can inhibit liver enzymes that metabolize drugs like warfarin and tacrolimus. For most patients, the evidence supports a safe, standardized dose, yet access to pharmaceutical-grade formulations remains a matter of geography, not science. Today, 450 human studies and decades of clinical use still cannot unlock a prescription pad in the United States. As legal frameworks outlast medical emergencies, the paradox endures. Life-saving evidence waits behind regulatory glass. Sometimes the gatekeepers shape medicine more than the healers. What do you think?